The paper we're going to discuss on this thread is Phosphorus Restriction as an Alternative to Chemical Plant Growth Retardants in Angelonia and New Guinea Impatiens from Horticulture, April 2018. The authors are Josh Henry, Ingram McCall, and Brian Whipker. It's a common practice in the nursery industry to apply plant growth retardants to bedding plants to keep them small and compact for sale. One of these, in fact, is paclobutrazol. It keeps the internode length smaller so the plants don't break as much, and they're easier to handle for the sellers. This also keeps them more attractive and more sellable. The problem with this practice is some plants are really sensitive to these plant growth retardants, or PGRs. They will lose buds before they can ever flower, or the leaves themselves will get really damaged looking, which makes them unsellable. In order to deal with this, what the authors of this paper did was they looked at applying nutrient regulation in place of these PGRs, namely phosphorus, in order to keep the internode length smaller. For this experiment, they used two different plants, Angelonia, which is not known to be damaged by PGRs, and New Guinea Impatiens, which does have a problem with PGR damage. They used 2 cultivars of each to rule out a cultivar issue. They took cuttings of each of these plants and placed them in a mix of 80% peat moss and 20% perlite. This mix kept all the nutrients out of the mix, and so they could completely regulate the phosphorus that was going into this. They took these cuttings, they put them on mist for 2 weeks, didn't treat them, didn't fertilize them. At that point, they transplanted them and started fertilizing them. Everything but phosphorus was held the same across the board. Phosphorus was done in treatments of 5, 10, 15, and 20 parts per million in drip irrigation. So the plants that would have grown faster at the 20 probably got even more fertilizer because they would have needed more water because they were larger. There were 16 plants of each cultivar. 8 plants of each cultivar from each treatment part, 5, 10, 15, and 20, were just treated with the phosphorus, and 8 from each were treated later with the paclobutrazol growth retardant. On the New Guinea Impatiens, they were given the paclobutrazol as a foliar feeding, 7.5 parts per million, at the 5-week mark. For the Angelonia, it was done as a drench, 4 inches per pot, at the 4-week mark, which are both standard inside of the nursery industry. They later measured height, diameter, number of branches, dry weight, and the growth index for each of these plants, and ran regressions on them. The control was a 20 part per million phosphorus with PGR added at the 4 or 5 week, depending on whether it was Angelonia or New Guinea Impatiens. They looked at the regression curve and the phosphorus height versus the PGR height. And what they could do is when they followed that curve from the PGR height to where the phosphorus height was, they could see how much phosphorus in parts per million got them that same effect. It varied a little bit cultivar to cultivar, but they managed to develop a range. It was 5 to 10 parts per million of phosphorus applied as a fertilizer would keep the plants at roughly the same height you would get with a PGR without adding it. If you go lower than that, you tend to get deficiency, and if you go higher than that, you didn't get the growth regulation that you wanted.